TALENs is also a class of DNA cleavage reagents, which were synthetically built from parts, conceptually similar to ZFNs. ZFNs were popular until 2010, but they were difficult to engineer, and editing outcomes from ZFNs are often suboptimal. In the ZFN video, I also discussed how ZFN modules have an overlapping influence on each other. TALENs actually solve a lot of the problems posed by ZFNs. TALENs came out in 2009. They are made up of tails, which are the programmable DNA binding domains first discovered in 1989. The second component of TALENs is the FOC1 nuclease domain. So it took around 20 years from the tail discovery to actually understand them enough to the point we could apply them for engineering. By the way, the FOC1 domain is exactly the same as we saw in the ZFN video, so I will not discuss it again. If you want details on it, check out the ZFN video. Instead, I want to focus on tails. Tails stand for Transcription Activator-Like Effectors, or TAL-effectors. And adding a nuclease to tails makes them TALENs. Tails are a class of proteins that were first discovered in a bacteria called Xanthomonas campestris. This bacteria infects the plant that produces peppers or capsicum. More specifically, this bacteria carries a plasmid that contains a gene that encodes for the tail protein. The tail protein is on the plasmid in the bacteria, yet it carries a nuclear localization signal. This was a surprising finding back in the 1990s because bacteria don't have a nucleus, so why carry a localization signal? A more important feature of this protein is that it contains a set of nearly identical repeat regions, and generally there are 15 to 20 such repeats. At the end of the protein is a transcription activator-like domain. Since the bacteria is a pathogen, it transfers this plasmid to the host using a specific secretion system. Once the plasmid is in the host uses the plasmid to synthesize the tail proteins from the gene. So the tail proteins are virulence-related proteins that are carried on the plasmid. Once the protein is made, the tails will go and bind to specific host genes, and specifically at the promoters of such genes. The important thing to note is that the DNA binding is done by this repetitive segment of tail proteins. The end or the C-terminal part of the protein has the activation domain, which can recruit RNA polymerase II onto these promoters. And this leads to upregulation of such host genes. This is the virulence outcome of bacterial infection. By the way, the N-terminal region of the protein has the NLS. And hopefully now you see the connection between activation domains and why they are called transcription effectors. Interestingly, in the tail proteins, if you take away everything except the DNA binding repeat region, the repeat region activity remains unchanged. It still binds DNA perfectly. But as a consequence of removing all other parts, it cannot activate any gene, meaning that it does not behave like a transcription activator. When making TALENs, it's only this specific repetitive segment that is used. All other parts are removed. So in TALENs, when you see that it is called a transcription activator-like effector, it does not actually have an activation function. It simply reflects that the repeats are derived from a protein that originally has that activation function. Since TALENs rely on these domains, let's try to understand how they function. As I said, they are nearly identical repeats, and each repeating monomer is about 34 amino acids in length. This monomer contains two helices, a short one at the N-terminal end and a long one at the C-terminal end. Amino acid residues 12, 13, and 14 are in the loop portion in between helices. The residue number 13 alone is responsible for base-specific contact with the DNA. And specifically, it binds or recognizes exactly one specific base. The binding is directional, meaning that the base lives on a strand which is parallel to the direction of the monomer. In contrast, if you remember, the DNA binding by ZFNs is actually anti-parallel. The residue number 12 has a major supporting role in determining the monomer stability upon DNA binding. On the other side, residues 14 to 17 are largely responsible for making sure that the monomer actually binds DNA and not RNA. Typically, residue number 12 is called repeat variable residue number 1. Number 13 is likewise the repeat variable residue number 2. Together, these two are called repeat variable diresidue. So, what I want you to take away from this is that there are 15 to 20 repeats of these 34 amino acid long monomer domains where only 2 amino acids, number 12 and 13, actually change. Every other amino acid, except this diresidue, is identical among repeats. This is because the residues determine base specificity, and it needs to vary if you want to recognize a new DNA base. To make this explicit, here's what I mean. This is the linear stretch of amino acids that makes up the monomer containing the helices. If you align all the amino acids for each of the repeat, for simplicity I'm only showing you 6 of the repeats, what you will find is that all the amino acids are exactly the same at every single position for every repeat, except for position 12 and 13, the diresidue position used for DNA binding. In 2009, the recognition code for diresidue DNA binding was eventually uncovered. If the diresidue was histidine and aspartic acid, the monomer recognizes a cytosine. If it's asparagine or isoleucine, it's A. Asparagine or glycine binds T. Asparagine and histidine bind G. Likewise, there are some other combinations that can also bind a specific DNA base. There are some diresidue combinations that recognize methylated cytosines, like the ones found on CpG islands. ZFNs, if you remember, have trouble binding the methylated DNA. Hopefully, tails are no longer a mystery. Now let's understand how they are used to build TALENs. So imagine this is some target DNA you want to cut. In this middle region, you can have any set of sequences, which is called the spacer, just like we saw with ZFNs. And we will circle back to this spacer in a moment. The DNA around this spacer is what you want to target using TALENs. The target binding is done by the tail repeats, and each circle here is one monomer that binds one specific DNA base, and these monomers are concatenated to form a chain. Since this is used for genome editing in eukaryotes, the N-terminal usually carries the nuclear localization signal. Just like ZFNs, the C-terminal end contains the FOC1 nuclease domain. The interesting bit here is that the first nucleotide is always a thymine, which is bound by 2 domains noted as -1 and the 0 position. Except for this first base, each DNA base is bound by exactly 1 domain. Now, this is not sufficient for DNA editing because FOC1 needs to dimerize for DNA cleavage. And just like the concept of ZFNs, you target the right side with a TALEN library as well. For TALENs in general, the spacer is quite longer. It is about 12 to 20 bases in length. And generally, you are trying to target about 18 bases on both left and right sides. I've only shown you 14 bases because drawing all 18 would be difficult to fit. In this, the DNA binding is always starting with a thymine for both the left and right side. It is bound by 2, sometimes 4, cryptic modules. I've only shown you 2 modules for simplicity. You can think of this thymine requirement at the 5' end as the requirement for the PAM in case of Cas9 enzyme. In meganucleases, this type of requirement is fulfilled by the central DNA bases. These modules have some basic residues or tryptophan that makes the sampling very specific to thymine. The aim of the 5' thymine recognition is to ensure that the rest of the binding gathers momentum. Essentially, the binding starts at the 5' at the T and then moves along the 3' end. This is like the PAM of Cas9 where PAM starts the sequence alignment. Another special feature of this recognition is that after the last nucleotide is recognized, which can be any nucleotide, there is a half monomer repeat. So instead of the typical 34 amino acids, it is only 17 amino acids. This half repeat is not involved in DNA binding. Its job is to add stability to the entire array. And because of this extra 17 amino acids, the FOC1 domain is located at a much further distance, which is why, compared to ZFNs, this spacer ends up being slightly longer. One last thing about this 5' thymine, because this position triggers the DNA binding process, mismatches in the 5' end between the tail RVD and its recognition base are not tolerated. Typically, 4 to 6 base mismatches can be tolerated out of a 20 base per target sequence, but those mismatches tend to be in the 3' end of the recognition sequence. Mismatches generally reduce TALEN efficiency because due to mismatches, the entire array is structurally disorganized. And for this reason, TALENs are more specific with target recognition than zinc finger nucleases. And for some targets, it's even better than Cas9. And as a consequence, they are generally less toxic. The biggest pain point in TALEN usage is that they are quite large. If you're targeting an 18 base pair region, then you have 18 tail arrays of 34 amino acid length, plus the half repeat, plus the NLS, and the nuclease domain. You need right and left side targeting as well, so all this is doubled in numbers. This totally ends up being in the 6 to 7 kb range. In comparison, meganucleases are less than 2 kb, ZFNs are about 2 kb, Cas9 is around 4.5 kb. The other issue with this large size is its assembly, primarily because tails are repeated structures, where the only variable portion is the diresidue in each binding monomer. If you have 18 tails, each tail is 34 amino acids, which means in total there will be only 36 amino acids that will be different. And assembling such repetitive sequences is not fun. The sad part about TALENs is that they were invented around 2009-2010. This was also the time when CRISPR-Cas9 came out, and so TALENs aren't really widely used. There are some very niche applications for TALENs. Tails can be attached to meganucleases, and these megatals can be very target-specific. Such applications and many others are similar to the offshoots that we saw in the case of ZFNs. In any case, I hope this video was useful, and as always, I'll see you in the next one.